In clinical oncology, identifying a single pathogenic mutation requires searching through hundreds of millions of DNA base pairs. The immediate challenge for a bioinformatician is distinguishing a genuine biological anomaly driving a tumor from a random optical error made by the sequencing machine. Modern bioinformatics pipelines, like Dragon, process this massive volume of data, but they do not use a single pass-fail quality threshold. This diagram illustrates a stratified quality matrix. To validate a mutation, data must pass three distinct checkpoints: base quality, alignment quality, and variant quality. These map chronologically: raw output in FASTQ, alignment in VAM, and final mutations in VCF. Validating raw sequencing reads for a clinical diagnosis requires mathematical proof at every single layer of this hierarchy. Memorizing terminology is insufficient. An analyst must understand how these metrics operate as sequential gates, filtering out digital noise to protect the final patient outcome. The process begins immediately after a sequencer, like the Illumina NextSeq 1000, outputs the raw data into a FASTQ file. The fundamental unit of this raw data is the read, a short fragment of DNA, usually between 75 and 250 base pairs in length. This graph charts the Fred quality score, assigning a confidence rating to every base read by the machine. Zooming in on Q30, the probability of an incorrect base call is 1 in 1,000, or 99.9% accuracy. Any data entering a clinical NGS pipeline must meet this baseline. Q30 is the clinical gold standard. High confidence in the base call does not mean the sequence is purely human DNA. During library preparation, artificial sequences called adapters are attached to the ends of the reads, which often degrade into low-quality tails. QC software tools like FastP or CutAdapt perform a process called trimming, slicing away these artificial adapters and degrading tails. If these artificial segments remain attached, the read will map to incorrect locations on the human genome during the next step. Base-level quality is the foundation. If corrupted or artificial data survives the FASTQ file, false mutations will inevitably exit the clinical report. Next, the cleaned FASTQ reads enter the alignment phase. Here they are mapped against a reference genome, like GRCH38, generating the BAM file. The first metric to check here is the mapping rate, the percentage of those millions of raw reads that successfully anchor to a valid position on the reference sequence. This animation illustrates how alignment quality is measured along two distinct axes: breadth and depth. Breadth of coverage measures the horizontal spread. What percentage of your targeted DNA region has at least one mapped read? Depth of coverage looks at the vertical stack, measuring how many times a base position was repeatedly confirmed. For standard germline testing, a depth of 100x is usually sufficient. Somatic cancer panels have different requirements. Tumors are heterogeneous, meaning they contain a complex mix of millions of healthy and mutated cells packed together. To detect a low-frequency mutation hiding within a tumor sample, clinical panels require depths ranging from 300x to 1000x. But high depth is only valuable if the reads are unique. During early preparation steps, the PCR process can copy a single DNA molecule multiple times, creating duplicate reads. Pipeline tools, such as Picard-marked duplicates, isolate and remove these identical clones to prevent them from artificially inflating the depth metrics. Base quality proves the DNA letter itself was read accurately. High mapping rates and genuine read depth prove that letter is in the exact biological location required to make a diagnosis. The software now moves to variant calling, scanning the aligned BAM file to isolate only the genetic anomalies, like single nucleotide variants and indels, and outputting them into a VCF file. This diagram shows variant allele frequency, or VAF. It is the percentage of reads at a specific position that carry a mutation compared to the normal reference sequence. Inherited germline variants typically present at roughly 50% VAF, indicating a heterozygous mutation. In somatic cancer testing, variants often appear at a VAF of 5% or even lower. This creates a critical mathematical dependency. The reliability of a variant allele frequency is determined by the total read depth at that position. These side-by-side charts show why. On the left, a 5% VAF at a depth of 1000x is supported by 50 independent reads confirming the mutation. On the right, that same 5% VAF at a low depth of 20x is supported by only a single read. Without the confirmation of multiple independent reads, A single mutant call is statistically indistinguishable from a random sequencing error. The variants in the VCF file are only as credible as the BAM file beneath them. A clinical variant is formally validated only when sufficient depth provides the statistical evidence needed to support its reported frequency. This dashboard shows how these mathematical thresholds materialize in an analyst's daily workflow using pipelines like Dragon. The metrics displayed here track the exact structural hierarchy we just mapped. Q30 verifies the base calls, mapping rate confirms the alignment, and mean depth ensures the variants have sufficient support. Once the software clears the data, analysts perform a manual review using tools like the Integrative Genomics Viewer, visually inspecting the alignments to ensure complex artifacts haven't slipped through. The final step is annotation. The structurally validated variant is cross-referenced against global databases like ClinVar and GNOMAD. Population frequency data from GNOMAD reveals if a variant is common in healthy individuals, while clinical reports from ClinVar help determine if it is a known disease driver. Only variants that survive this entire gauntlet are assigned an official ACMG classification, ranging from benign up to pathogenic. Clinical NGS data analysis functions as a chain of structural validation. By understanding how each layer of data protects the next, analysts ensure that the final pathogenic diagnosis driving a patient's targeted therapy is always a rigorous, evidence-based conclusion.